Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntac Consultants in Oakland, California, and the coordinator of the SERDP and ESTCP webinar series. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to remediate 1,4-dioxane at military facilities. First, Dr. Jerry Schnorr and Dr. Reed Simmer from the University of Iowa will talk about the use of phytoremediation for the treatment of 1,4-dioxane and chlorinated solvent co-contaminants at DoD sites. Second, Ms. Caitlin Bell from Arcadis will discuss bench scale testing to configure commercially available membranes for 1,4-dioxane bioreactor treatment. Each of the two presentations will be followed by a brief Q&A session, and depending on how we're doing on time, we will conclude the webinar with a longer Q&A session. The next two slides provide instructions on optimizing your webinar experience. If you have not done so already, download Zoom at the link shown here and provide it to you in your webinar registration confirmation email. And if you cannot download Zoom, you can still view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you continue having difficulties or if your screen freezes, just try keying in Control and F5 to perform a hard refresh of your screen. Keep in mind that you can download a PDF of the slides from the webinar webpage. That's available right now. We'll drop a link in the chat to the webinar webpage so you can download a PDF of the slides. You can then call into the conference line provided to you in your webinar confirmation email. One last viewing option, we are live streaming the webinar on the SERDP ESTCP YouTube channel at the link shown on the slide. Today's broadcast will be listen only. You can submit questions using the Q&A box on your screen. You do not need to wait until the Q&A period to get your questions in. We do encourage you to submit them well in advance of the Q&A sessions. We do ask that when you submit your questions to add your organization name at the end of the question so that we can identify it during the Q&A. Please do not submit questions in the chat box because the chat box is reserved for comments related to technical difficulties. And with that, I would like to introduce Dr. Hunter Anderson, who joined SERDP and ESTCP last year as a subject matter expert in the environmental restoration program area. And prior to that, Hunter provided technical support for the Air Force Civil Engineer Center since 2010 in the areas of emerging contaminants, environmental engineering, environmental toxicology, and quantitative data analyses. Hunter received his doctoral degree in environmental chemistry from the Ohio State University. Hunter, thanks for joining us, and please get us started.
All right, thanks, Rilla. Welcome, everyone. So yeah, just a quick series of introductory remarks here from the program office, particularly for you folks that are new to the webinar series. So yeah, SERDP and ESTCP are the DoD's flagship environmental RTD&E programs. SERDP invests heavily in basic and applied research that sustain and advance operations at our facilities. SERDP supports industry, federal agencies, and largely universities in every stage of research and development. SERDP's sister program, the Environmental Security Technology Certification Program, or ESTCP, is where we take these innovative technologies developed under SERDP and field them at scale to demonstrate and validate those technologies for an unbiased cost and performance analysis to supply our decision makers at installations and facilities with the information they need to adopt these technologies at scale. SERDP and ESTCP are both housed under the Office of the Assistant Secretary of Defense for Energy Installations and Environment, EI&E. And in doing so, that means we work hand in glove with all the other components within DoD to broadcast this information and to disseminate these innovative capabilities to the field where they're needed. Our projects are directly in alignment with national security priorities and provide the data and capabilities that DoD leadership need to shape guidance and instruction aimed at securing our military installations and facilities. This collaboration ensures policy decisions encourage and facilitate the adoption of scientific approaches, methodologies, and technologies that advance the DoD mission. With more than 320 installations in the portfolio, we, of course, span a very diverse range of geographies and challenges across the country. For this reason, our portfolio matches the breadth and scope of these issues. Our science and technologies protect built and natural infrastructure, strengthen DoD chemical and material resilience, and ensure the health and safety of our warfighters. Innovation investments also drive significant time and cost savings, resulting in a return on investment in critical mission needs. Our research portfolio is organized across what we call 6 program areas. The Environmental Restoration Program, which is the focus of today's webinar, is focused on managing PFAS and other emerging chemicals of concern in the environment. The Resource Conservation and Resilience Program area focuses on sustaining the 27 million acres of installation and natural infrastructure for test and training ranges, as well as critical nearshore environments or environmental security threats to the military mission globally. The Weapons System and Platforms Program area focuses on reducing the environmental impact and improving the chemical and material resilience of weapons systems and platforms operations and maintenance. The Installation, Energy, and Water Program area demonstrates and validates technologies that enhance resilience against disruptions to public energy and water infrastructure. The Natural Hazards Adaptation Program area demonstrates innovative and cost-effective technologies and methodologies to ensure that DoD installations can withstand and recover from natural disasters such as earthquakes, hurricanes, floods, and wildfires. Finally, the Munitions Response Program area is focused on unexploded ordnance in the underwater environment, including ponds, lakes, rivers, estuaries, and coastal ocean areas. After years of successful investment, this program area is currently transitioning technologies to end users through a series of test sites and live site demonstrations. So SERDP and ESTCP are a critical funding source and partner for hundreds of federal universities and industry R&D efforts. Our investments link our defense and industrial bases to the broader economic and innovation ecosystems within the country. We release several solicitations throughout the year. Researchers from the federal government, private industry, and research institutions alike can apply for SERDP and ESTCP funding opportunities. To receive updates about funding opportunities, program updates, and upcoming webinars, you can sign up for our mailing list at the link on this slide. Our webinar series highlights outstanding projects across all the various research portfolios. You can find additional information about the upcoming webinars at this link. All past webinars, additionally, are archived and can be accessed at this link as well. I would like to remind you that a copy of the presentation of today's session can be downloaded from the webinar webpage. We would appreciate it if you could please take a moment to complete the survey that will pop up at the end of this broadcast to facilitate our constant improvement efforts. At this point, I'll turn it back to you, Rula, to introduce our speakers. Thank you.
Thank you so much, Hunter. It is now my pleasure to introduce the speakers for today's first technical presentation. We'll have 2 speakers. The first one will be Dr. Jerry Schnoor, who's the Allen S. Henry Chair in Engineering and professor in the Departments of Civil and Environmental Engineering and Occupational and Environmental Health at the University of Iowa in Iowa City. Jerry is also the co-director of the Center for Global and Regional Environmental Research. He has served as PI and co-PI on projects funded by SERDP and ESTCP, the National Science Foundation, NIEHS, and the US EPA involving 1,4-dioxane, polychlorinated biphenyls, chlorinated solvents, nanoparticles, and explosive compounds. Jerry is a member of the U.S. National Academy of Engineering, elected in 1999 for research and engineering leadership in the development, validation, and utilization of mathematical models for global environmental decision-making. He received a bachelor's degree in chemical engineering from Iowa State University, a master's degree in environmental health engineering from the University of Texas at Austin, and a doctoral degree in civil engineering, also from the University of Texas at Austin. The second speaker for our first presentation is Dr. Reed Simmer, who is a research scientist at the University of Iowa, where he has worked with Jerry since 2016. Reed's current research focuses on the bioremediation and phytoremediation of groundwater contaminated with 1,4-dioxane. Since 2022, Reed has served as a lead research scientist on the ESTCP project that you'll be hearing about today. He is also the co-PI on a SERDP project that began in 2024, focusing on developing innovative strategies to improve the survival of a metabolic dioxane degrading strain for field bioaugmentation. Reed received a master's degree in geography and geographic information systems and master's and doctoral degrees in environmental engineering, all from the University of Iowa. We're delighted to have you both. And Jerry, I'm going to ask you to please go ahead and get started.
Well, thanks, Rula, very much. And welcome, everyone. I'm Jerry Schnorr, and with me is Reed Simmer here at the University of Iowa. And as stated, we would like to tell you about bioaugmenting phytoremediation in order to treat 1,4-dioxane contaminated groundwater. I'll be responsible for telling you the technology background and our demonstration at a DoD site, the overview and design. And Reed will be responsible in this presentation outline for the last few bullets, including systems performance, conclusions, and next steps, and the benefits to the Department of Defense as a result of this novel technology. 1,4-dioxane, as you know, it's a widespread groundwater contaminant in the U.S. The map, which is drawn on the right side of this particular slide, shows all the locations of public water systems that are in excess of desirable concentrations for 1,4-dioxane. So people are drinking 1,4-dioxane right now, even as we speak. That's of concern because the larger are these circles, the orange and red circles, the larger they are, the higher the concentration. And it goes up to 34 micrograms per liter, mostly in groundwater systems. Now, EPA, if you do a risk estimate, 1 in a million cancer risk for 1,4-dioxane, you get a health advisory level of around 0.35 micrograms per liter. That means a difficult treatment option in that you have to get down to very, very low health advisory levels in treating this problem, but also the plumes are rather dilute in the beginning on the order of 100 micrograms per liter or even less. In technology overview of our option, that is phytoremediation plus bioaugmentation, we use a metabolic degrader, a bacterial degrader, that degrades 1,4-dioxane very fast. And it's used as a sole carbon and energy source in this case. So there's not much food in the form of 1,4-dioxane, but it can degrade it rather completely. If you have a spill in a groundwater system, such as depicted in this slide, you might have chlorinated solvents, including TCE, co-mixed with 1,4-dioxane quite often. Now, we know from our previous research and others that the phytoremediation will work mostly for these chlorinated solvents. For example, TCE can be degraded quite readily in the root zone of the trees or grasses, both aerobically and anaerobically by natural bacteria in the system. Now, due to the proper lipophilicity of TCE, some of it may be taken up into the plants, and we know that there are enzymes, cytochrome P450s and monooxygenase enzymes, which can degrade these chlorinated solvents. In the case of TCE, we can even monitor for metabolites in the plant tissues, such as trichloroacetic acid shown here. Nevertheless, some may escape into the atmosphere, shown here as TCE, and it is degraded in the atmosphere readily by hydroxyl radicals with a half-life on the order of a couple of days. Now, the 1,4-dioxane, however, is a bit more complicated story because it's not readily degraded naturally in the root zone of vegetation. And therefore, we recommend bioaugmenting and making a good habitat, even a biofilm for bioaugmenting organisms in the root zone, which can oxidize 1,4-dioxane, even mineralize it all the way, and thus decreasing the amount of 1,4-D taken up into the plants. Nonetheless, the plants can metabolize 1,4-dioxane. We know that, and we can measure the metabolites. Some may escape in the transpiration stream, as shown here in this schematic. As a technology overview, our particular organism that we're using now is Rhodococcus ruber 219, again, a metabolic aerobic degrader of 1,4-dioxane. As shown here in the photo, it grows very handsomely, bright orange and happily on 1,4-dioxane in this enrichment culture, especially when supplemented with thiamine, that is vitamin B1. It goes very fast. In fact, compared to other organisms which are reported in the literature as metabolic degraders of 1,4-dioxane, it's much, much superior. As shown in the last blue line of this table, it has a fast substrate utilization rate and a very low half saturation constant Ks of 0.015 milligrams per liter. And that results in an S-min, the concentration of dioxane that's necessary to sustain the culture of a very, very low concentration, say 0.49 micrograms per liter, highlighted in the orange circle here. That means that even dilute plumes, say less than 100 micrograms per liter, which is typical, can take the dioxane concentration down to health advisory levels. That's what we're after. Now, our demonstration is at the former Twin Cities Army Ammunition Plant, TCAP, in Arden Hills, Minnesota, just north of Minneapolis and St. Paul. And our demonstration site is shown in map view here with a 4,000-acre former TCAP site. And in particular, there's many contaminations, but we're working at Site G in the orange circle near the bottom, near the south of the Twin Cities Army Ammunition Plant. If you look at the groundwater plume at that Site G location, it's shown in cross-section here. And looking into that plume, the groundwater table is around 875 feet above sea level here. And the water table is the blue line shown here across. And you can see the plume of TCE in orange contours in cross-section, and also that of 1,4-dioxane co-mixed in green contours. The 1,4-dioxane ranged when we started this project from 10 to about 500 micrograms per liter, and the TCE was 100 to 5,000 micrograms per liter. The entire plume was discovered in 2016 at the site, and Site G in the right-hand slide shown here, it seemed to be running off in the south and west direction. It runs off of the TCAP property line under the highways. And in fact, on the left, you can see it goes maybe 7 or 8 miles down to the south, inundating the source water for 2 towns, New Brighton and St. Anthony. And those have had to go to point source treatment of advanced oxidation in order to still use that groundwater source. What we want to do is try to treat the contaminant zone at Site G in order to clean up the source. And for that, we chose phyto-attached growth reactors in consort with Ecolotree Incorporated, our subcontractor at the site. Now, they've been successful in using tree boxes, tree planters, such as shown here on the left, in order to treat municipal wastewater and also to treat landfill leachate. But we have modified that design to what we call now Fager 2.0, and it's fully automized, as Reed will tell you about, in which we've put screened PVC monitoring wells. Of course, we recommend bio-augmenting the root zone of those plants, and that's all enclosed in 75-gallon HDPE containers now, such that it's above ground and easy to get a mass balance. And of course, no more worry about discharging and making a new spill at the existing DoD site. In design, we are pumping the groundwater up into these tote reservoirs shown on the left, and heading to the right, a total of 40 Fager reactors shown with 2 trains, 4 replicates in each on each side, 5 rows total, 5 rows times 4 replicates times 2 parallel trains equals 40 reactors in the current system. It's also shown in schematic with a water balance here. The Fagers are irrigated then with 50 gallons per day of groundwater, a split stream off of that recovery system that we showed in cross-section, and 50 gallons per day roughly is transpired as 10 gallons per day from each row of the system, and that leaves 0 gallons per day for discharge from the 5th row. And that is how we operated the entire summer of 2024, with 0 liquid discharge, the only possible discharge being any transpiration from the plants of TCE and 1,4-dioxane. Now the figures are bio-augmented. As we said, each and every one is identical. 4 replicates, as mentioned earlier, planted with willow and poplar trees and bio-augmented with Rhodococcus ruber 219. In addition, another R modification to the system is to have a dosing pump feeding vitamin B1 so that we have very rapid degradation rates, bright orange, happy bugs, as shown previously. We monitor the water table, and as the water table gets low enough, we pump remotely controlled pump into each of the series of reactors. In the spring of 2023, this is how it looked at the TCAP site of Twin Cities Army Ammunition Plant. We had 24 Fager reactors at that time with 2-year-old poplar and willow cuttings growing actively, and that was installed in 2023. In 2024, it grew to 40. We added more reactors, 40 Fager reactors in series, as shown earlier. And this is how it looked in the spring of 2024. And then as the plants leaf out, this is the way it's shown in spring of 2020. They're now 3-year-old cuttings of willow and poplar. And moving on, Reed is here to tell you about how that was operated, the demonstration, pilot demonstration, the automation of it, and its performance. Reed?
Yeah, thanks, Jerry. I'm going to talk about some of the techniques we used to monitor and control this system remotely. So all of the figures were irrigated from these HDPE irrigation pipes through drip manifolds that are 8-way manifolds, and then through drip spikes that go about 4 to 5 inches below the surface. And that way we're applying the contaminants directly to the root zone and directly to where we bio-augmented. We used sonic water level meters that you can see on the left here at the top of a screened well that just thread in, and the sonic water level meters measure the water level every 3 to 5 seconds. And when the water level reaches a minimum, we program these to switch on the irrigation pump and then water each row from the previous row in series until it reaches a maximum point that we control as well. And in that way, irrigation is driven by water demand by the trees. And that way trees never run dry. And then we also don't overwhelm the system as well. And we also use Arduino microcontrollers to measure the water level every 15 seconds. And via a remote controlled laptop, we were able to monitor this remotely from back here in Iowa. And then also I could modify our desired water table to try to change different settings. And everything was housed in a job box here with solar fans. So it was weatherproof, it was secure, and the fans kept all the electronics cool. And here you can see our Rhodococcus rubra that was grown in a Sartorius fermenter, 30-liter fermenter. And each phager received about 1 liter of cells, as you can see here, that we applied just onto the surface. We sampled the system every 2 weeks from May through August, the last 2 years, from these composite bottles that it's a flow-through bottle between each row. And that way we always have the most current sample possible when we go to sample. It's not been sitting there for an extended period of time. And we also have valves at the bottom of the effluent manifold if we need to take another sample from the very bottom of those phagers. And we use peristaltic pumps, as you can see here. And to capture DNA that may have washed out, the Rhodococcus rubra DNA, we filter about 1 liter through the SteriVex filter, as you can see here. And we use that for quantitative PCR. We also sample in VOAs in triplicate to measure 1,4-dioxane and chlorinated solvents. Now transitioning to our results. You can see here, this is a plot of the THMA, DXMA copies per mil in the effluent that was captured on those SteriVex filters. And this is a metric of the cell population of Rhodococcus rubra that we added. The THMA DMXA gene is the gene that is used to degrade 1,4-dioxane, and that is why we monitor it. As you can see, the population stayed the highest in the first stage here because stage 1 saw the highest 1,4-dioxane concentration. And then in series, in general, moving down, we see a decrease in the Rhodococcus rubra population. However, throughout the 9-week sampling period shown here, it was quite stable, which indicates us that we were able to maintain a high Rhodococcus rubra population despite low influent dioxane concentrations. Now looking at the dioxane concentration through time, you can see with this line here what was coming in the influent. And then in general, just as seen with the gene copies, the inverse is true here. We see a decreasing dioxane concentration through time and through each stage, dropping all the way down to about 0.5 microgram per liter at the lowest. So we were able to reach over 90% of treatment by the end of the treatment system throughout this 9-week sampling period. We did see a little bit of drop off towards the end, and this may indicate that bioaugmentation is necessary to maintain treatment. Now looking at TCE, TCE was between 10 and 30 microgram per liter, and it was effectively treated by our system, again, decreasing through the treatment stages all the way down to almost below detection. Similarly, cis-DCE, we saw it was effectively treated, again, to well below the MCL and almost to below detection, below 1 microgram or 0.1 microgram per liter. 1,1,1 trichloroethane, again, similar story. We did see a little bit of a spike here where it was not as treated as well in the early stages, but by the end of the system, it was treated again to near below detection. We did see some minor vinyl chloride formation that we attribute to anaerobic conditions in the root zone, but it was removed by to near below detection and well below the MCL by this stage 4 mark here. But this is something that we need to monitor in terms of keeping the water level low to ensure that aerobic conditions. In conclusion, the phytoremediation system bioaugmented with Rhodococcus ruber was capable of sustaining remediation of dilute 1,4-dioxane to as low as 0.5 microgram per liter, and we were able to maintain a stable population of Rhodococcus ruber for 9 weeks. That population was highest again in the first treatment stage, but it may be that repeated bioaugmentation is needed to maintain extended treatment. Poplar and willow trees were capable of complete transpiration, so we were running 0 discharge. No installation-derived waste was generated whatsoever. And this also means that any breakthrough that may have happened, it did not happen, but if it did, it would have been removed. The mass would have been removed effectively by transpiration. Again, chlorinated solvents were also treated to below MCLs. Our next steps, we are processing some core samples that we took that you can see here on the right, very densely packed with roots with those 3-year-old poplar and willow trees. And we'll be doing some DNA analyses here to see biofilm formation. We're also looking at phytoforensics to look at, with Joel Birkin at Missouri Science and Technology, to look at uptake of 1,4-dioxane and chlorinated solvents, if any, into the wood tissue, to just look and ensure that we're getting adequate treatment in the root zone. We're also looking at 1,4-dioxane metabolite analyses. These metabolites are very short-lived, especially in a root zone, and so it's very challenging to measure them at low concentrations, but that is direct evidence of metabolism. So we are doing our best to measure those low concentrations. We're also very interested in pursuing full-scale implementation of follow-on project if any opportunities arise. Wrapping up, benefits to DoD. Phytoremediation is a cost-effective, low-carbon, aesthetically pleasing, sustainable technology. It's well suited for large dilute plumes like 1,4-dioxane and chlorinated solvents. And again, the trees are capable of transpiring all the water applied, so we can run at 0 discharge. Phytoremediation coupled with bioaugmentation, the root zone is well suited for microbes that grade 1,4-dioxane. The trees pull in mobile contaminants like 1,4-dioxane and bring this to this bioactive zone that we've bioaugmented and engineered. The microbes are then able to form biofilms on the roots, and as the roots grow, the biofilms come with it, and that way we're expanding the treatment area. The roots also release dissolved oxygen and root exudates to propel metabolism of 1,4-dioxane. It's suitable for using in the ground as well with pump and treat or in above ground reactors. In addition, the Fager technology is very interesting for small footprint, tight areas. It's a modular design. You can bring these on site. The trees are already established. The root zones are ready to go. You can add microbes ahead of time, and you can do proof of concept and treatability studies as close to full scale as possible with these Fager setups. And then the Rhodococcus rubra 219 strain that we've been researching here for a number of years is effective as a bioaugmentation strain for phytoremediation, but it's also highly promising as a standalone metabolic strain. And we are looking at that in a current SERDP project as well. And with that, I'll wrap everything up.
Great. Thank you so much, Jerry and Reed, for a great presentation. And a reminder to everyone, if you have questions, please get them into the Q&A box. And for additional information on this project, you can click on the hyperlink with the project number or enter it on the SERDP and ESTCP webpage. All right, so we've got a few questions and I'm going to relay to Jerry and to Reed. The first one is from the Michigan Environment, Great Lakes and Energy. Can rainwater or seasonal changes create any issues with the reactors and their efficacy?
Well, thanks for that question. Of course, the rainwater varies across the country. Hybrid poplar and willow can work just about anywhere in the United States, even in Alaska and down to the subtropics. But if you have a lot of rainfall, that has to be treated in an open system, as we've shown you. And it has been and can be in Minnesota, as we've shown. Wintertime operation is more tricky. We can talk about that, but right now we're not operating in winter. Rather, we'd be pumping the groundwater in the summertime and transpiring it. And then as the groundwater plume moves in the wintertime, we'd pump it back up in the summertime. If it was a drought condition, then that's not a problem, because then the wastewater can be increased in order to treat even more water as a result of drought conditions.
Thank you, Jerry. This next question is from the California Department of Toxic Substances Control. Did you use a specific soil to grow the plants? Did you mix it with compost? And did you observe any difficulties or challenges with increasing salinity in the later stages of the reactors?
So we actually used a perlite material that's very lightweight and inert in this system, and that was very helpful in this above ground reactors because we're able to move them around, and it also allowed for excellent drainage. So certainly adding soil to the system would complicate things in terms of, TOC or salinity. But in this case, we did not have any issues with that because we were using inert perlite. That is the white popcorny material that you find in potting soil, and it's a volcanic rock product.
Great, thank you, Reed. This next question is from the Colorado Department of Public Health and Environment. What was the total volume of groundwater treated during the 9-week 40-unit pilot test?
We treated a slipstream. I didn't have time to tell about it, but the entire Site G groundwater plume is being captured by those 4 pumps with screens shown in the previous slide. And most of that, 90+% of it, is going to an advanced oxidation treatment system because of the fact that already groundwater supplies of nearby towns has been impacted. But ours is a demonstration pilot project, taking a small slipstream, and we were treating about 50 gallons per day as shown, just a small portion of the total capture zone that is being pumped up.
Great, thank you so much, Jerry. This next question is from the TRS group. How would a full-scale system look? And any idea of the maximum GPM per acre and maximum water concentrations that a system like this could handle at full scale versus pilot scale?
We estimate that if we had been asked to or allowed to plant a plantation of trees at the site, they have 4,000 acres, there's plenty of room. We would have needed about 200 acres to plant that, to take care of 250 gallons per minute using drip irrigation. Now that's with the climatology of Minneapolis, St. Paul area, but I hope that gives the questioner some idea. We can scale up, but it would be with a large plantation of trees where you have a good sized area with which to work.
Great, thank you so much, Jerry. This next question is from the Central Truckee Meadows Remediation District in Nevada. To what depth below groundwater, below ground surface, is effective treatment accomplished? So how deep can you go?
If it's just phytoremediation alone without pump and treat, we can go about 15 feet deep. But this example we just showed you into the planter boxes, and you can always use pump and treat onto a plantation of trees. But the example we just showed you was 75 feet below ground surface.
Thank you, Jerry. And this next question is from the Department of the Navy. And it has to do with whether any of the leaves that may fall off the trees are sort of captured and disposed of if they contain 1,4-dioxane.
Yeah, we do not see 1,4-dioxane accumulate in the wood tissue. It mainly remains in the transpiration stream. So it, about 80% of it has been shown to go directly to the atmosphere. And if it does stay in the wood tissue, it's broken down by P450 monooxygenases, similar to our liver enzymes that phytoremediation's often referred to as the green liver. This is the same mechanism. So fortunately, with 1,4-dioxane, we are not concerned about losing 1,4-dioxane through the leaves that are lost.
But I would add, thank you, Reed. I would add to that when you bioaugment, the beauty of that is the rapid degradation of 1,4-dioxane by, in our case, Rhodococcus ruber, greatly decreases that transpiration stream. And we're actually still analyzing the leaf samples at Missouri Science and Technology, Joel Birkin. So I don't have an exact answer for what fraction is going into the air right now, but we suspect it's much, much, much less due to the biodegradation, and the enzymes are involved. We know that there's biodegradation occurring in the system, in the root zone.
Great, thank you both, Jerry and Reed. And a follow-up question for clarity. So what is the ultimate fate of TCE and 1,4-dioxane that is transpired to the atmosphere?
It's degraded, oxidized by hydroxyl radicals, the cleansing agent of the atmosphere, and the ultimate product is chloride, carbon dioxide, and water.
Wonderful. Thank you for that. Following up on the scale-up question, can you reiterate whether there are any scale-up issues associated with the culture? Is it a fast-growing culture? How well does it survive in the subsurface environment at the concentrations of contaminants of interest?
It's a very easy culture to work with in terms of scaling up. It grows so rapidly on 1,4-dioxane. So that way you're able to keep those enzymes active. We have a current SERDP project with Rice University looking at ways to increase the survival once you bioaugment it into the subsurface, especially because of the vitamin limitation. We're working on ways to deliver vitamins more effectively and increase survival in that way. So we feel that it's a very scalable technology, and this microbe can do well in full-scale bioaugmentation systems.
Great. Well, thank you both for a fantastic presentation and a very engaging Q&A session. We still have questions that are coming in, and we'll get to them at the end of the presentation. So if you could please stick around. We're gonna shift gears to our second and last presentation that will be given by Ms. Caitlin Bell. Caitlin is a 1,4-dioxane technical expert at Arcadis in Seattle. She has nearly 20 years of experience in site investigation and remediation, and since 2015, she has served as the team lead for 1,4-dioxane for Arcadis in North America, where she supports 1,4-dioxane projects across the board. She also serves as a technical lead on a variety of projects at federal and commercial facilities, and was an author and editor of the Emerging Contaminants Handbook, which was published in 2019 and included chapters on 1,4-dioxane and other emerging contaminants. Caitlin served as a member of the ITRC team that produced the 1,4-dioxane technical guidance document, and as part of the ongoing training team. She received a bachelor's degree in chemical engineering and a master's degree in environmental engineering, with her master's being obtained from the University of Illinois at Urbana-Champaign. Caitlin, we're delighted to have you. Please get started with your presentation.
Great. Thank you so much, Rula, for that introduction. I appreciate it. Happy to be here today to chat a little bit about using membrane biofilm reactors for ex situ treatment of 1,4-dioxane. So another sort of approach to bioremediation, and here we're focusing on the microbial bioremediation side of things. So just to get things started, we'll touch a little bit on some groundwater treatment technologies for 1,4-dioxane and see how this fits into the toolbox. We'll dig in a little bit into 1,4-dioxane bioremediation, just so that everyone has a sense of metabolic versus co-metabolic bioremediation. We'll talk a little bit about membrane biofilm reactors and why they're attractive for this sort of treatment, and then get into the ESTCP project overview and some of the bench scale configuration efforts that have been going on, wrapping up with our conclusions and some of the benefits that we think will be available to DoD. So why are we here? Why are we looking at this? It's because 1,4-dioxane is commonly occurring with chlorinated solvent plumes. There are a lot of historical sites that have legacy pump and treat systems for treatment of those chlorinated solvents, using things like air stripping or activated carbon to effectively remove those chlorinated solvents from the groundwater. However, those treatment technologies are not particularly effective for 1,4-dioxane. And so many times this 1,4-dioxane plume remains in the subsurface untreated and can grow larger while the chlorinated solvent plume is shrinking. So air stripping isn't particularly effective for 1,4-dioxane treatment because 1,4-dioxane isn't particularly volatile. And large-scale activated carbon isn't particularly useful for 1,4-dioxane because it doesn't absorb very well to carbon. So what are some options for groundwater treatment for 1,4-dioxane? On the ex situ side of the house, we're looking at things like advanced oxidation processes, or AOPs. We're also talking about some synthetic media that are available on the market that are more designed to absorb that 1,4-dioxane to it than GAC. We're also looking at bioreactors, which will certainly be the topic of discussion here today. And then sometimes some folks are able to utilize small scale activated carbon at, say, a wellhead treatment option or a residential treatment option when they're able to sort of change out that carbon in a more frequent manner. On the in situ side of the house, there's things like in situ chemical oxidation, or ISCO. There's thermal treatment, as well as phytoremediation, which we just heard quite a bit about here today. And then, of course, bioremediation and its association with natural attenuation. So when we're talking about 1,4-dioxane biodegradation, we could be talking about either metabolism or co-metabolism of that 1,4-dioxane. So normally when we think about biodegradation, say for chlorinated solvents or for petroleum hydrocarbons, we're talking about metabolism. Those microorganisms are utilizing those compounds for generating energy, for creating new biomass, and for living out their lives. And there are microorganisms that can metabolically degrade 1,4-dioxane. We just talked about one of those in the prior presentation, and I'll touch on one of those here in a little bit that I'll call inoculum 1. So when you hear that again later, that's a metabolic biodegrading organism. However, we're also focusing here quite a bit on co-metabolism of 1,4-dioxane. And when we're looking at co-metabolism, the microorganisms are actually using a different primary substrate. So in this case, we're going to talk a lot about propane. And they're doing their energy generation and their biomass growth from utilizing that propane. However, the enzymes that they generate as part of those ongoing processes can also go on to degrade 1,4-dioxane to oxygen and carbon dioxide, innocuous end products. So we're leveraging this sort of fortuitous side reaction of co-metabolism to help broaden the spectrum of concentrations of 1,4-dioxane that we can treat, of microorganisms that we can use, and approaches that we can take in order to biodegrade 1,4-dioxane. So if we take a closer look at one of the in situ applications of co-metabolic biodegradation of 1,4-dioxane, we can look at something like propane biosparge. So in this case, we're injecting things like air and propane into the subsurface, along with sometimes a bioaugmentation culture that can use that propane if they're not already there naturally. Also injection of some nutrients, so that doesn't become a limiting factor. And there are projects that have shown that this propane biosparging approach is effective at degrading 1,4-dioxane down to regulatory levels. So then on the other side of the house, why are we thinking about membrane biofilm reactors, or MBFRs? Why not look at other bioreactor configurations that are out there? There are other projects and other work by others that are looking at those sorts of things, like fluidized bed reactors. But some of the nice things about the membrane biofilm reactors are shown here on this slide. On the one hand, they're bubble free. So essentially the gas is delivered via diffusion through the membrane. So just to describe it a little bit more, in the graphic you see on the right-hand side, there's sort of these sheets of the membranes which then get wound around together, and you can deliver gas or liquid sort of through the middle of the membranes, and the gas in particular will diffuse through the membrane and out toward where the biofilm is. So that diffusion is a nice characteristic of these membrane biofilm reactors because it helps deliver just the right amount of things like propane to the microorganisms. It's also a highly efficient process. So rather than having to bubble propane in water, whereas some of it might get let off to the atmosphere, here we can deliver it through these membranes in a nearly 100% efficient manner. They're also pretty flexible. For example, we could deliver just propane through the membrane. We could deliver propane and oxygen through the membrane. We could deliver just oxygen through the membrane. So it gives us some options for ways to configure these bioreactors when we get to the field. And lastly, these membrane biofilm reactors generate really robust biofilms. So the microorganisms are able to adhere to the outside of those membranes. They're propane is right there, their oxygen is right there, all of the things that they need in order to grow and proliferate are right there with them at the surface of the membrane, and so it makes for a really good working biofilm. And so what we're looking to do here are bring these technologies together. So utilize what has been learned from full-scale field implementation of things like that propane biosparge at a couple of sites out in California and in Michigan, as well as bringing these membrane biofilm reactors that have been used historically for things like perchlorate and nitrate reduction. So they've been used by delivering hydrogen through those membranes rather than propane. So really what we want to do here is configure these membrane biofilm reactors to deliver propane and oxygen, and then demonstrate whether this is an effective means for ex situ co-metabolic biodegradation of 1,4-dioxane. So our objectives here is to start with that configuration of the existing membrane biofilm technology. That's what I'll talk about more here in a little bit, as well as looking to optimize those membrane biofilm reactors for the field demonstration. So both the configuration and the optimization were done at the bench scale and leading into the later field demonstration for this year. That field demonstration will be to evaluate the overall efficacy of this and really to quantify some of those cost savings and sustainability benefits. So we want to compare these membrane biofilm reactors to things like the advanced oxidation processes that are kind of the gold standard at the moment. So we want to bring a more sustainable option to the table, hopefully also something that is more cost effective since it won't have a lot of the energy requirements that a lot of those AOPs have. So with the configuration steps that were completed at the bench scale, they started with some gas permeation tests, mostly just to see how the propane and the oxygen move through the membrane. And then we had some bench scale membrane biofilm reactors set up, did some proof of concept testing, and then looked to start with that optimization. So really it's a matter of dosing the best ratio of propane and 1,4-dioxane into those membrane biofilm reactors so that we can effectively degrade the 1,4-dioxane while also minimizing the amount of propane that's needed. So if we take a closer look at some of these gas permeation tests, here we're looking at the oxygen test. Very simply at the bench scale, you put a bit of this membrane into a container, deliver 100% membrane through, or sorry, 100% oxygen through the membrane, and then see how much of that oxygen comes out of the membrane, passes through the membrane. And generally, this is a pressure-related relationship. So the higher the gas pressure delivered inside the membrane, the more oxygen will pass through the membrane. And so the graph on the right shows that there was a nice linear relationship between that, and we now have an understanding of how much oxygen would need to be delivered inside the membrane in order to see effective presence of it outside of the membrane. If we take a look at the propane permeation test results, so same idea as the oxygen, except this time the propane is going through that membrane and passing into that container, and we're measuring the amount of propane that comes through. We actually tested two different membranes. CP is for composite membrane and PP is for polypropylene membrane. And in summary, both of these membranes deliver propane effectively. We found that the composite membrane actually allowed more propane to pass through. However, when we start thinking about scale up, that propane membrane, or sorry, that polypropylene membrane also allows an appropriate amount of propane to pass through it. So both of these can be options on the table for scaling up. Then if we take a look at the membrane biofilm reactor setup at the bench scale, on the left, you can see those white strings. Those are the little bits of the membrane. You can also see some biomass growth on those white strains. So we know that the microbes can establish their biomass on that membrane. And on the right-hand side, you can see the setup. Get my cursor here. The bioreactors are here in the middle, and then there are a series of pumps and sampling ports associated with sort of circulating the water that has 1,4-dioxane in it. So getting into our proof of concept testing. So here we started with very high 1,4-dioxane concentrations, 60 milligram per liter, 1 milligram per liter. We used a bioaugmentation culture that metabolically degrades 1,4-dioxane, so that inoculum 1 that I was talking about earlier. And we only added propane, or sorry, we only added oxygen. We did not add propane. And what you can see from the graphs on the right-hand side, the top bar shows the oxygen delivery. The middle bar is really where the action is. It shows the influent 1,4-dioxane concentrations in the black squares and the effluent 1,4-dioxane concentrations in the red circles. And you can see that there was effective removal of the 1,4-dioxane, about 93% in that first stage when we started with 60 milligram per liter, and about 95% removal in that second stage when we started with the 1 milligram per liter. And then getting into that optimization that I was talking about, so looking at the different propane to 1,4-dioxane mole ratios. So a similar graph here as before. The top bar shows oxygen and propane delivery. And then the next 3 bars show 1,4-dioxane removal based on different bioaugmentation cultures. So here again, we have inoculum 1 that we're taking a look at. And then on the bottom, you see there's a mark there for ENV425. So this is a Rhodococcus ruber strain where we're focusing on its ability to use propane to co-metabolically degrade 1,4-dioxane. And then in the middle, we actually had a mixture of the two of those. And so here we are delivering both oxygen and propane. We have various ratios of propane to 1,4-dioxane. You can see those kind of across the top. And the take-home message here is that we did see 99% removal of 1,4-dioxane starting at some lower concentrations, so about 100 micrograms per liter, so something a bit more environmentally relevant, down to below 1 microgram per liter, which is our detection limit at that time. And with this data, we were able to hone in on sort of the most effective propane to 1,4-dioxane ratio to start with from the field demonstration. So just a couple of takeaways from the MBFR configuration step. When it comes to membranes, that polypropylene membrane is going to be adequate at the large scale. That's the one that was selected to be moved forward, partly because that's the one that is used in those existing commercially available membrane biofilm reactors that we were looking at before, those ones that have been used for perchlorate and nitrate in the past. We were also able to hone in on that mole ratio and plan on starting by having a propane to 1,4-dioxane mole ratio of about 6,000 to 1. That should allow us to facilitate a healthy, robust biofilm that will degrade 1,4-dioxane effectively, while also not over-delivering propane because there can be some substrate inhibition. If the microorganisms start getting too much propane, then the enzymes don't want to go on to react with the 1,4-dioxane. On the bioaugmentation side of things, we found that ENV 425 is effective. That bioaugmentation culture is commercially available, so that will be moved on to the field demonstration. And we did notice that we need to give that microorganisms quite a bit of time to acclimate in the bioreactor. So we'll be taking that lesson learned into the field as we start to get things set up out there. And then when it comes to oxygen delivery, we found that oxygen could be delivered through the membrane. However, for sort of logistical purposes, we've decided to deliver the oxygen into the water and just deliver the propane through the membrane. That'll keep things a bit more simplistic and still provide the microorganisms with both the propane and the oxygen that they need. So now we're moving into the field demonstration portion of the project. The site selected is at Arnold Air Force Base. This is a site that has 1,4-dioxane and groundwater at about 30 micrograms per liter, as well as some chlorinated solvents with TCE at about 55 micrograms per liter. There is an existing pump and treat system with an air stripper in place. So those chlorinated solvents are already being removed. We can take a slipstream either before or after that air stripper and be able to run that slipstream of water through our membrane biofilm reactor. And we're actually coordinating with another ESTCP project. It's a different configuration of bioreactor so that we can see side by side how these two perform in the field. So our field demonstration includes several different steps along the way. We're currently in this setup step. We're working with the folks at APT Water to configure their existing membrane biofilm reactor to meet the needs of propane delivery, of oxygen delivery, of the nutrient delivery that we want to have as part of this system. So once that gets delivered out to the site, we'll be seeding that system with the Rhodococcus ruber ENV 425, so that one that utilizes propane and can co-metabolically degrade 1,4-dioxane. We'll be dosing that up with propane, oxygen, nutrients, trying to get that biomass established on the membrane so that when we flip the switch and start the flow-through portion of the testing, those microorganisms will be ready to start degrading 1,4-dioxane. So at the start, we're going to take a slipstream from that existing system after the air stripper so that we can focus just on 1,4-dioxane removal and things like propane utilization and begin to optimize the different portions of the system. And then we'll move into the challenge portion of the testing where we'll take the slipstream from before the air stripper. So now there'll be some chlorinated solvents in here as well. We want to see if those chlorinated solvents would also be removed from this co-metabolic mechanism. And then we'll be running a series of tests, challenge tests. So starvation tests, removing the oxygen from the system for periods of time, seeing if the microorganisms can bounce back, changing up the propane purity a little bit so that we can see if we can use something like a residential grade propane or whether there are impurities in there that might be inhibitory. So just trying to challenge the system a little bit, understand where the limitations are, and find the optimized configuration for that 1,4-dioxane removal. And then just for a bit of a visual, this is conceptually what the system will look like. Those two tall tanks in the middle are where those membrane biofilm reactor modules will be dropped in. So on the figure that we looked at earlier, where those membranes were wound around a central post, each of those can be put into a larger tank. So here we'll have, say, 4 of those modules in each of our tanks. And that's a good way to be able to scale up for full scale for this system in the future. You can just have more tanks with more modules. We also will have a compressed air component to the system for delivering that air into the water before it goes into the membrane biofilm reactor tank. And we also have a nutrient dosing system so that we can make sure we don't end up limited for things like nitrate and phosphate. So in conclusion, so far we've seen that these membrane biofilm reactors can achieve 1,4-dioxane reductions down to below 1 microgram per liter. That bench scale testing that was completed provided some critical design parameters for going to the field demonstration. That design for the field demonstration is complete and are just working on getting that equipment together and out to Arnold Air Force Base. So we are hoping to complete that field demonstration portion over the course of 2025 here. And then just to round things out, a reminder of some of the benefits to DoD. We're looking to address this unreliable removal of chlorinated solvents and 1,4-dioxane concurrently from these existing legacy pump and treat systems that have things like air strippers or activated carbon on them. So these membrane biofilm reactors are meant to provide another option for augmenting, say, existing pump and treat systems if something needs to be added on to remove the 1,4-dioxane, or for new systems that might be going in the future. The nice thing about these membrane biofilm reactors is we are providing a destructive mechanism for treatment of 1,4-dioxane. It's not just transferring it to activated carbon. It is actually destroying the 1,4-dioxane itself. And we're looking to identify this as a cost-effective and sustainable option compared to things like AOP. Some of those details will certainly be borne out after the field demonstration. And with that, I'll turn it back over to you, Rula. Thank you.
Great, thank you for a wonderful presentation. Again, for additional question opportunities, just drop in your questions in the Q&A box on your screen. We did receive a few questions, Caitlin, that I'm going to go ahead and relay to you. The first one has to do with whether the propane delivery was pulsed during any portion of the study to induce cyclical starvation of propane. Was that the case at all?
Yes, absolutely. I believe that question's likely in reference to the in situ propane biosparge projects that I was talking about. There, we find that it's helpful to cycle that propane on and off. That way, those enzymes have a chance to go on to destroy the 1,4-dioxane rather than the microorganisms just getting this constant stream of propane, so they just want to grow more and more and more. So there is a balance there between providing enough propane for these microorganisms to grow, but also creating those starvation conditions, those pseudo-starvation conditions where those enzymes can degrade the 1,4-dioxane. So we also are looking to bring that to the membrane biofilm reactors. That's why we were looking so closely at that propane to 1,4-dioxane ratio, so that we can find that sweet spot where we're providing enough propane that they can grow, but not so much that the 1,4-dioxane doesn't degrade.
Great, thank you. This next question is from the Oregon Department of Environmental Quality. What is the planned square footage and flow rate for the full-scale setup that you're going to be doing or demonstrating in 2025?
Sure. The field demonstration system, I want to say it's maybe it's in a Connex box. So maybe say a 10-foot by maybe 20-foot tops sort of footprint. Everything will be housed within that Connex box, the membrane biofilm reactor tanks, the dosing, the nutrient dosing system, the air compressor, the controls and various things that are needed. As for flow rate, we are looking at maybe only 5 gallons per minute, for example, of what will be sort of taken from the main treatment system and diverted to our membrane biofilm reactor system for evaluation.
Thank you, Caitlin. This next question is from the Denver Department of Health and Environment. Can the membrane biofilm reactors effectively reduce 1,4-dioxane from the tens of microgram per liter concentration levels to below the health risk thresholds that Jerry talked about?
Yes, we believe that they can. One of the nice things about co-metabolism is that it's fairly agnostic about your starting 1,4-dioxane concentration. So that's why it can be really helpful for these types of situations where we have low starting 1,4-dioxane concentrations and still need to get to, say, 0.35 micrograms per liter or 0.3 if you're in various states for their cleanup levels. So I think that will be borne out certainly as part of the field demonstration, but we are anticipating that this type of co-metabolic approach can negotiate those types of sites that have low starting concentrations, as well as sites that have higher 1,4-dioxane starting concentrations. It can kind of run the full gamut.
Thank you, Caitlin. This next question is from Haley and Aldrich. What is the hydraulic retention time that was used for the laboratory testing for the membrane bioreactor?
Oh, I'd have to go back and double check. I want to say off the top of my head, I want to say it's maybe on the order of 4 hours, or at least that's the number I believe that we're carrying over into the field.
Great. Thank you so much. Is the laboratory data summarized in a report on your project website on the SERDP ESTCP project website by any chance?
Yes, I believe so. It was all reported out in sort of a treatability study report, and we can verify that that is posted up to the website.
Great. Maybe if we can go back to slide 71. This is the project number. If you guys drop it in the search bar for the SERDP ESTCP on the SERDP ESTCP website, it should take you to the project webpage where a lot of excellent resources are saved. All right, another question for you, Caitlin. Can metal precipitation from groundwater be a challenge as far as the biofilm functioning?
Sure, that's a good question. Certainly when we're adding things like oxygen, changing redox conditions, metals precipitation can be a thing that needs to be considered. Certainly that's a site-by-site sort of discussion and evaluation. What's nice about what we're doing out at Arnold Air Force Base, because they already have an existing treatment system, it actually already includes air sparging, so there's lots of air being passed through that groundwater already. They've already accounted for accommodation for any needs of metals removal or bag filters up front, all of that sort of stuff. So we'll be able to take our slipstream of groundwater after all of those components as our starting point for testing the membrane biofilm reactors and can just focus on that part of the technology.
Great, thank you. Do you happen to have any initial estimates on what the cost savings might be for using the technology described as compared to, for example, AOPs?
We have run some initial numbers. And for example, I want to say membrane biofilm reactors, for when it comes to capital cost, could be maybe 40% less than AOPs might be, especially a UV-based AOP. And then when we're talking about O&M type of costs, maybe something like a 15 to 20% reduction when we're looking at those membrane biofilm reactors. So those initial values are very, very promising, and we're hoping that it will be collecting the data as part of the field demonstration to help firm those up and do a more robust comparison.
Thank you. And one last question before we pull Jerry and Reed back into the discussion here. Can you talk a little bit more about the time needed for acclimation and potential impacts once you scale up the system related to acclimation?
Sure, absolutely. So, having an acclimation period for any sort of bioreactor is not uncommon, right? So if it's a fluidized bed reactor, if it's a membrane biofilm reactor, if it's a moving bed bioreactor, if it's at say a wastewater treatment plant, or if it's treating something like perchlorate at a groundwater remediation site, there's generally going to be kind of an acclimation period while that biomass proliferates, establishes itself, and essentially takes hold on the parts of the bioreactor where it's going to live. I've seen that be on the order of, say, 3 weeks to 3 months, depending on the type of bioreactor, the microorganisms, the situation, the temperature, all of that sort of thing. So we are planning for, I think, a several week acclimation period for this field demonstration. Certainly we'll be gathering information about how long is actually needed for that and what that means for scale up. But yeah, I think it's generally a practice in the world of bioreactors, and it just depends on the configuration and the site conditions of how long that may take at full scale.
Thank you, Caitlin. That's a really thoughtful response. And thank you again for a great presentation. We have 2 questions that have come in for Jerry and Reed that remain unanswered, and then but please stay on. I'll have one more question for you. But Jerry and Reed, can you please talk about how much vitamin B1 was used during the field test? This is a question from Haley and Aldrich.
Sure. We just use a small dosing pump. When we grow them in the lab, we only need about 50 ppb vitamin B1. We overshot that with our dose because of scavenging from native organisms. The other challenge is that vitamin B1 is very unstable, especially at higher pHs. So we had to use a buffer to make sure the pH stayed low, especially when the vitamin stocks were exposed to the elements. They're also very heat sensitive and light sensitive. So again, we have that SERDP project to address some of those constraints. But it's not a ton of vitamin B1, but more of the challenge is the stability of vitamin B1.
Thank you, Reed. And this last question, how do you know when it is safe to shut off the pumps and shut down the system, the treatment system?
Thanks for that question. We measure the influent concentration from the pump and treat continuously. So this is the age-old question of rebound, and that is you would turn off the pumps when you've reached the agreed upon record of decision for the treatment level required. In our case, in Minnesota, it's less than 1 microgram per liter for dioxane. But the TCE might be a problem with rebound. It's at very, very low concentrations, as we showed you. But after you turn off the pumps, assuming that 1,4-dioxane would control that decision, if there was rebound of TCE afterwards due to microglobules of DNAPL or whatever that could be present at your site, you should leave the phytoremediation system intact so that you could restart at some point if you needed to. I don't believe that would be true in our case because there doesn't seem to be any residual DNAPL present. But that is, of course, a concern always when you're talking about when can you turn off the pumps of a pump and treat.
Thank you so much, Jerry. Any last thoughts you want to leave our audience with, especially as they pertain to next steps and when the data would be available from these planned next steps for your project?
Well, we'd love to move this to some other site if anybody out there is interested, because this is the last year. Now, we are doing a site demonstration in July of 2025, but after that, we'd like to move this somewhere else. We think it's a good technology and we'd like to try it elsewhere. However, also, one of the things that we'd like to try is a plantation of trees, you know, much more natural system, and pump and treat. There you'd have to have lysimeters underneath the system to show that indeed you're not contaminating any groundwater supplies with the remainder that percolates back to the groundwater.
Great, thank you, Jerry and Reed. And Caitlin, any sort of last thoughts that you want to reinforce before we wrap up here?
Oh, I might just mention that I'm very excited to see these membrane biofilm reactors get deployed into the field, and I look forward to talking with folks in the future about the results from the field demonstration. Thanks so much.
Great. Well, thank you all for a wonderful webinar. I would like to wrap up by reminding everyone that these webinars happen every other week. So our next webinar is on Thursday, February 13. It will focus on DoD-funded research efforts to implement thermal microgrids at DoD installations. This webinar will feature two speakers, Dr. Michael Wetter from the Lawrence Berkeley National Lab and Dr. Amy Allen from the National Renewable Energy Lab. Please visit the SERDP and ESTCP webinar webpage to register for this and other upcoming webinars. And like Hunter mentioned, you can also access archived webinars from the last 10 years and listen to those recordings if interested. The audio and a copy of the presentations of today's session should be available shortly on our webinar webpage in case you would like to refer to it. And at this point, we'd like to thank you and ask you to please take a moment to complete the short survey that will pop up on your screen. This concludes today's webcast. Thank you for joining us.